Hey guys, I'm going to pull up first thing first all of the learning goals for this video, and there's a whole list. These are all of our goals, and we're going to kind of take a weird winding route through this list. So we're going to be talking about meiosis and how it gives us genetic variation. We're going to review mitosis a little bit and see what it has to do with meiosis and where the differences lie. And we're also going to review a little bit about pedigrees, Punnett squares, chi-square, linked genes, and the difference between, say, Down syndrome and Tay-Sachs disease. Okay, so step one, we're going to look at mitosis versus meiosis, and then study some really unique things about meiosis that make life the way that it is.
Let's zoom in on this diagram here. So mitosis is something that you're very familiar with. We know all of the stages of interphase, G1, S, and G2. So by the end of interphase, a cell that's about to go through mitosis has replicated DNA, and that's why this chromosome here is shaped like an X, instead of just one little bean. We also have double the organelles, we have room to grow, and all the resources that we need. In mitosis, your chromosomes line up. Here we have 2 chromosomes and 4 sister chromatids, and then they separate to create 2 identical daughter cells. I tried to make these as identical as I could. I realized this one looks like half dark, half light, but these are supposed to be 2 identical daughter cells that are made because of mitosis.
Now let's check out meiosis. What's similar is that interphase is pretty much exactly the same. So we're going to have G1, S, and G2. We have twice as much DNA as we normally would. That's why this chromosome also looks like an X. So in both scenarios, interphase was similar, and in prophase, our chromatin has condensed into chromosomes. Here's where things get different. In prophase 1, we have this thing called synapsis. It can also be called crossing over, and it's when it looks like these chromosomes, they do kind of this stanky leg dance, and the homologous pair can sort of mix and match itself. It's as if one of these is a zebra, the other one is a horse, and now here we have a horse with a zebra tail, here we have a zebra with a horse tail. So here would be the results of one cell after prophase 1 of meiosis. You can see we have some recombinant chromosomes here that have pieces of others, pieces of the other one in its homologous pair. So imagine half your DNA is from mom and half is from dad. It's like saying, here's your mom chromosome, here's your dad chromosome. They swapped some alleles back and forth. So now the ones that you're going to pass on, they're not necessarily going to be 100% the ones you got from mom. Let's see these 2 sperms here that were produced by the end of meiosis 2. In this sperm, we have one chromosome that's pretty much been left untouched by crossing over, and another one that is a recombinant chromosome. So it's not entirely from dad or entirely from mom. It has this brand new combination of genes that we've never seen before. So now that we've seen some differences between mitosis and meiosis, I want to remind you why meiosis is particularly important. An example of variation is, for example, in a human family where there are 10 offspring. There's very, very low chances that 2 of those offspring will look exactly the same unless they're identical twins. Because when eggs and sperm are being made through meiosis, you can produce so many combinations of eggs and so many different combinations of sperm. So we're going to go ahead and list, now that we've looked at meiosis, what are 3 ways that meiosis gives us so much variation in offspring. It makes everyone look completely different from everybody else. First thing that's going to produce variation, chromosomes do that stanky leg. So I'm talking about crossing over in prophase 1. So let's draw a picture of crossing over. This creates variation because now it's possible to inherit a unique combination of alleles from mom and alleles from dad. So this is during synapsis. These chromosomes are in prophase 1 of meiosis. Another way that eggs and sperm have infinite different variations of genetic material that can be held inside of them is chromosomes assort independently, which means they kind of randomly go to one side or another of the cell during metaphase and then are pulled apart in these unique patterns. So I'm going to draw out an example cell, and we're going to watch all of the ways that independent assortment can provide variation. So number 2, we can cause variation through independent assortment, or the random distribution of chromosomes. Let's say that we have an original cell that has already gone through meiosis 1, and here are the chromosomes inside of it. We already have some recombinants in here.
So during the second cycle of meiosis, the eggs and sperm that are produced have a lot of different options, even if we're all starting from the same cell that's finished with meiosis 1. So let's see independent assortment in action. One set that we could draw of separated sister chromatids is the one on your left, and the other one on your left. Here's a chromosome. Here's another chromosome. The next way to do it, we're going to take those other pairs that we haven't used yet. This is one option for 2 eggs or sperm that could be produced from this cell, but there are at least 3 more. It's just like foiling. So we're going to draw just one more option. So this is option 1. I don't want to take much of your time. And let's get option 2 going. This is like a whole other world. Different scenario. We take left and right, and then right and left. So this chromosome could randomly be selected, along with this one. And then in our second sperm, the other 2 could be independently assorted into it. So now, the sperm that we're creating are very unique from one another because of these random events. Third random event, and the last one that has to do with reproduction, is the actual moment of fertilization itself. If you're a female, imagine that you have ovaries with hundreds of different egg cells, each one genetically distinct. If you're a male, you produce hundreds of different sperm cells, and each one of those is genetically unique because of crossing over and independent assortment. So whenever we fertilize, we're taking a random egg and a random sperm. The chances of these 2 being similar to eggs and sperm of the past are very, very low. And when they combine their genes, they are just mixing up the genome even more and adding to the variation in our species and in the species of plants and other animals.
There's one other source of variation, and I'm going to pick a different color for this. The funny part here is that these things don't happen in every organism on Earth at all. This doesn't happen in bacteria. Bacteria have circular chromosomes, they reproduce through binary fission. They're not going through this whole independent assortment meiosis process, but they will go through one other process, and that is mutation. So in purple here, mutation is our 4th source of genetic variation, and this happens in every single living organism that contains DNA on Earth. It can even happen in non-living organisms like viruses. So mutation is going to happen to all organisms.
You will absolutely, on the test, see a multiple choice question or a free response that asks you either which of these happens in all organisms, or asks you to list 3 things that happen in meiosis that contribute to variation, and you will know exactly what to do. We have already accomplished learning goal A, which is figuring out the 4 sources of genetic variation, and we've already accomplished C, which is comparing and contrasting meiosis and mitosis. I'm going to do one more diagram that helps us figure out meiosis versus mitosis after erasing the board.
If you see a diagram, how will you know if it's meiosis or mitosis? One easy way is count the chromosomes that you started with and that you end with.
Mitosis is how we take diploid body cells, or 2N cells, and create more diploid cells. In other words, you're taking a cell with 46 chromosomes in 23 pairs, and you're going to copy every single chromosome over and split your cell exactly in half just one time to produce an identical cell from that. So both of the daughter cells are identical in mitosis.
Let's get funky on the other side, and let's write meiosis. Meiosis starts off much the same way with one 2N body cell. So the first cell in the process has 46 chromosomes. It's going to produce not 2 daughter cells, but 4 unique daughter cells. And each one of them will be 1N. So not only do these cells have just 23 chromosomes each, they are unique and genetically distinct from each other because of the things that we talked about before. Not mutation, but because of crossing over, which only happens during meiosis, and because of independent assortment, which only happens during meiosis. In mitosis, you have 2 completely identical copies. Now that I've told you the perfect meiosis scenario, we're going to look at what happens when we mess this up. The making of eggs and sperm gets a little wonky.
We're going to end up with cells that are all genetically unique from each other, but there will be some mistakes made that will either remove a chromosome accidentally or add a chromosome accidentally to an offspring. So I'm telling you that we're about to talk about non-disjunction.
So we're mentioning moments when chromosomes do not separate or disjoin as they should during meiosis. I'm going to talk about what that might cause, and then we'll talk about the origin of non-disjunction. So you're familiar already with these 3 different diseases, at least the first one. First disorder that can be caused by non-disjunction is called Down syndrome. I'm just going to write DS. And it's not just any extra chromosome that can cause Down syndrome. There's one specific chromosome, and it's number 21. And you're born with Down syndrome if you're born with 3 copies of chromosome 21.
Most people have 2 copies. Only people with Down syndrome have 3 copies. How does that happen? Well, it must be true that the cell from one parent had 2 copies, and the cell from the other parent had 1 copy. 2 plus 1 made 3, and for one parent to have 1 copy and the other one to have 2, something had to get messed up because those should be perfectly even.
The next disease we're going to talk about, these 2 are to do with the sex chromosomes, so that 23rd chromosome. We have Klinefelter's syndrome. What that would look like is if someone had 2 copies of X and only 1 copy of Y. I've drawn the Y chromosome really tiny because it actually is physically like really, really small. So this person's genotype would be described as XXY because they have 2 X's and 1 Y. They're biologically male. They may have gotten 2 X's from mom and a Y from dad. They may have gotten 1 X from mom and an XY from dad, depending on who had non-disjunction happen during meiosis. The last disease that we're going to talk about, right on this side, is Turner's, and this one happens to the ladies. Turner syndrome is when you have no Y chromosomes, so you're biologically female, but you do have 1 X chromosome instead of the usual 2. So their genotype would just say X. So they don't have an extra like Klinefelter's or Down syndrome. They have one that's missing. If you hold on a second, I'm going to draw some pictures of non-disjunction. This cell has 22. This cell has 22. One of them gets both the X and the Y. One of them gets nothing. So now this cell can produce only sperm that have just 22 chromosomes where they should have 23, and there are no sex chromosomes present in the sperm.
Let's take this cell and find our final products of meiosis 2. We can have 22 autosomes, an X and a Y, 22 autosomes, an X and a Y.
That means let's take a normal egg cell here that has 22 autosomes and an X. If the egg combines with this sperm, we're going to get a cell that has 44 chromosomes and then an X. So this person has Turner syndrome. They have 1 sex chromosome and it's an X. If we combine the egg cell with this sperm cell, this person has 44 autosomes, an X, and a Y. Here's where I got that. I took the X from mom and then the XY from dad, and I added them all together. So here is someone who has Klinefelter's syndrome. We are on to bigger and better things, so on our to-do list, I just need to show you how phenotypes can be different because of the environment, what linked genes are, and the difference between chromosome disorders and gene disorders. So we've already looked at chromosome disorders and how they're caused. They're caused by non-disjunction. We're going to look at gene disorders next. All gene disorders are caused by one thing, and it's not non-disjunction. They're caused by a mutation. That mutation can be on any autosome or the sex chromosome, and it can be dominant or recessive. So I'm going to take a colorful marker, and we're going to draw a family tree of someone with a dominant disorder. One example of a dominant disorder is Huntington's. It's on the autosomes, so I'm not going to draw any X's or Y's. I'm just going to have a family where it's possible, but not probable, that mom and dad both have the dominant gene for Huntington's, but they carry the healthy gene for not having the disorder. And they have a daughter who's homozygous recessive and will never develop Huntington's. She inherited 2 alleles that did not have the gene mutation on them. Next, let's switch colors, and we'll draw Tay-Sachs and sickle cell anemia. This gene disorder is especially common in people whose heritage is from Israel and other parts of the Middle East. If 2 parents have Tay-Sachs, then their children must also have it, because this is an autosomal recessive disorder. So if we fill in the genotypes, we could write little t's for Tay-Sachs, little t's for Tay-Sachs. Now, of course, you probably will not see this family tree in reality because 2 parents would not make it to their reproductive age if they had Tay-Sachs disease. So let's see 2 carriers have a daughter with Tay-Sachs. 2 heterozygous parents have their homozygous recessive daughter. When we switch colors, we're going to draw another recessive disease. This is the last one, and it has something extra super cool about it. So this one is for sickle cell anemia. We'll just call it sickle cell. It is a recessive disease, so let's write recessive on the bottom of these 2. So I'm going to draw a family tree that looks just the same because 2 carriers can pass it on to their son or their daughter. Let's have carriers. I'm going to use the letter S even though it's forbidden. I'm so sorry, guys. The one unique thing about sickle cell anemia, and the reason that I'd like to mention it, is that there's actually an advantage to being a carrier in this situation. Sickle cell anemia is most common in people who have African heritage because if you're heterozygous for sickle cell anemia, you happen to have increased immunity to malaria, that disease carried by mosquitoes. Kyle, you're in the video. We're really working on this list here. The only thing that we have left to do is look at phenotypes due to the environment and linked genes, and that's the most fun part of it.
I'm just going to tell you something you already know, which is there are some plants that if you put them in acidic soil, they turn one color, and if you put them in basic soil, they turn a totally different color. So you could put 2 genetically identical plants in 2 different soils and see 2 different phenotypes. So you'll see some questions about that, about how if different genotypes are always the cause of different phenotypes. The answer is no. For example, animals that live in the Arctic, like the Arctic hare, will be white during the winter and then brown during the summer. Those phenotype differences aren't because its chromosomes changed. It's phenotype change because of input from the environment. Another freaky example is going to be sex determination in salamanders. I'm just going to send you a video about that and you're going to mind blown. The last part of this video is linked genes, and it's the tough part. Okay, get ready. I'm going to show you normal life that you thought was real, and then I'm going to show you linked genes. Okay, back to meiosis, when chromosomes can do the stanky leg. Here's 2 homologous chromosomes in the very beginning. One is from mom, one is from dad. But when you pass these on and make your sperm and your egg cells, it's not true that you will pass on these same chromosomes that you got from mom and dad, because they're going to recombine with each other. Here are chromosomes that you could actually pass on to your offspring, and you could tell that some of them are called recombinants. Here's a recombinant chromosome.
well, it's a chromatid right now, but it will soon be split into 2 different chromosomes. Now, these genes, G and A, are not called linked genes because they're very far apart from each other. They're on the same chromosome, but chances are when synapsis occurs, they're going to be separated by that. It's not too crazy that they would end up mixing and matching on different chromosomes. This diagram looks the same, but it's a little different. Here we have genes G, B, and A. There are 2 different alleles of every gene. When crossing over occurs, genes that are far away from each other are likely to be separated, but genes that are really close together probably won't get separated. So, for example, if we look at B and A, they're such close neighbors that they probably won't be separated even if crossing over occurs because they're just too close together. The chances of synapsis happening right here at this location in between the 2 is really, really low. That's why the other day we looked at pictures of people with red hair and freckles. Those are linked genes because they're so close together on the chromosome. They're usually inherited together. Very rarely are they inherited separately. It's so hard for the body to produce a chromosome that has the recessive versions of those genes. Oh my God! All I have left to do is show you some linked genes practice problems, and I just really hope that you enjoy yourself while you're watching this. One thing they could do is give you a set of genes and say, how close to each other are they on the chromosome? So, example problem. A chromosome has 4 genes on it, A, B, C, and D. We're going to get some data on how often they recombine or have crossing over events in between them. And that's going to tell us who is closest to who. So here's the data you would get as part of the question.
It would tell you B and D cross over 6% of the time, C and A cross over 14% of the time, A and B cross over 31% of the time, C and D cross over 44% of the time, C and D cross over 50% of the time. So these are pairs of genes, and this is the frequency that they might recombine or cross over.
The way you work this out is basically just a logic problem, nothing too scary. I like to draw a big chromosome, it's like a noodle. The genes that recombine the most often are the furthest away. So C and D have to be on the ends because they recombine more than any other pair of genes. So if crossing over happens anywhere on this chromosome, it's going to separate C and D because they're the furthest apart. Now, let's look at who is really, really close together. B and D are hard to separate. They're only going to separate 6% of the time. That tells me that I can put gene B very close to gene D. Okay, we only have one more gene to put on our chromosome, so kind of goes without saying that we're going to end up putting gene A here. So the answer to this problem might be that the genes are in the following order, or of course you could write it backwards and say D, B, A, C. We are going to do 2 more of these problems because yes, you're going to see them on your test. So here are all of the genes that are on one chromosome. We just don't know who is closest to who. The data that we're getting this time is not how often they recombine, but their distance. And remember, the further they are apart, the more often they should be separated from each other by crossing over at the chiasma during synapsis. Here's our data. When we draw our chromosome, let's use that same logic again. We want to find the largest distance. That's between H and G, so I'm going to put those the furthest apart that I can. It doesn't matter which one goes on the top, which one goes on the bottom. I just did that because I felt like it. Now I want to look at, okay, well, who's the closest? That's A and G. They're only 2 units apart, so I'm going to put A right next to G. My last number is that B and H are also pretty close together. They're only 8 units apart. So I can go ahead and put my last gene there. So my answer is H bag, or you could write it GABA. That's the order of order of genes on this chromosome. Here comes another chromosome with genes A, B, C, and D. So the genes, this will be a great time to pause your video, do this on a slice of paper, and then check back for the answer. Really. Pause your video. Here comes your answer. The furthest apart genes should recombine the most often, so B and D recombine or are separated the most often, so let's put those on opposite ends. Then the closest ones are B and C, so I'm going to put C next to B. Finally, we have A left to put in here, and A is very close to D. Our answer is BCAD. You could also read it DACB. If you got one of those, you're good. You have one final skill to learn. The skill is looking at the results of a cross of fruit flies and seeing if the genes that we're thinking about and modeling with our cross, are they linked or are they not linked? So this is a problem that basically has two answer choices.
Either the two genes are linked, aka they're next door neighbors, they're really close to each other on a chromosome, or the genes are not linked.
They're on separate chromosomes or the same one but very far apart. Mendel assumed a lot of things. There were his 3 laws. First law was some genes are dominant. We're not going to break that rule. The next law is some chromosomes assort independently and that they always segregate. Well, we're never going to have 2 chromosomes travel together unless there's non-disjunction. We're not breaking the law of segregation here. We're going to break the law of independent assortment if genes are linked. So I'm going to write independent assortment, and I'm going to cross it off because if the genes are linked, we'll see this law broken. Here's what that looks like in a problem. These are almost always about fruit flies, and here's what it'll look like. This is the dominant allele for red eyes. This is the dominant allele for white eyes. Recessive. Here's the dominant allele for normal wings, and here is the recessive allele for vestigial wings. I'm just going to write vest. They're going to say, we did a test cross, here are the results. Your test cross is going to be a heterozygous fly crossed with a fly that's homozygous recessive for both traits. The reason why this is a great thing to work with is twofold. One of them, we can make predictions really quickly.
There's a chance of a quarter for every single phenotype. What I'm saying is, if we foil this guy, first, outer, inner, last. And we foil this guy, first, outer, inner, last. All of these possible gametes are identical. So, there are only 4 possible identities of the offspring.
Here's another way that this is really strong science. Take this fly. We're going to assume something about his parents. We're going to assume we know he's heterozygous because one of his parents was homozygous dominant and the other one was homozygous recessive. That means, theoretically, if Gregor Mendel was correct, this heterozygous fly's chromosomes
should look like this. And one of them came from dad and had the dominant gene and the dominant gene, and one of them came from mom, had the recessive allele and the recessive allele.
If these genes are on the same chromosome, some other funky stuff's going to happen. I'm going to erase these so we can draw what might be happening if the genes are linked. If the genes are linked, then they have to be on the same chromosome. So, we have one copy from dad and one from mom, and in this case, dad had the following genotype, and mom had the following genotype. Okay, so when this fruit fly goes through meiosis and makes 4 gametes and gives them away, if these genes are linked, we will most often get chromosomes identical to what it already had from its parents, because these are not going to be recombined in unique combinations.
So let's highlight the gametes that are going to be really common. Your common gametes are homozygous dominant and homozygous recessive. This is what happens if the genes are linked. Your results are going to look wild and crazy. So let's say there are 368 flies of this genotype, 336 flies of this genotype, and then 56, and then 40.
We have clearly broken Mendel's rules because these are not even numbers of a quarter, a quarter, a quarter, a quarter of the total. Instead, it's much more common to have these grandparent phenotypes because these genes are linked and they're very close together on a chromosome, and meiosis is not causing the same amount of variation as it usually does. I'm going to do another problem with y'all, but a lot neater. We're going to start with the data and end with a decision about linked or not linked. Let's talk about dogs. In dogs,
short hair is dominant and long hair is recessive. Black paw pads are dominant and brown paw pads are recessive.
let's talk about a test cross where we had a heterozygous dog and a homozygous recessive dog. If Gregor Mendel is right and the genes are not linked, we expect the following.
A quarter of the dogs to have short hair and black paws. A quarter to have long hair and brown paws. A quarter to have short hair and brown paws. And our last quarter is our last phenotype, which is long hair and black paws.
This is our prediction. If this turns out to be true, the genes are not linked because independent assortment and crossing over took care of variation in the way we think it will. Let's check our data. For the following phenotypes, we have the following offspring.
105, 95, 103, 97.
We can assume that these are pretty even looking, and that means that the gene for coat length and paw pads is not linked. Gregor Mendel was right. These genes are not close together on the chromosome. You could do a chi-square test about this, and if your predictions match reality, then the genes are not linked. If your predictions do not match, something is wrong, and the genes are in fact linked. So let's look at one of those. Let's go back to fruit flies. We're doing a test cross as usual. I don't know, wrinkled wings, red eyes is what they always talk about. Remember that hypothetically, this heterozygote had two homozygous parents. So if these genes are in fact on the same chromosome, he's got one with strictly dominant alleles from dad, one with strictly recessive alleles from mom. That's because no matter how many times mama crossed over, she had chromosomes that were identical to each other. So, even though some crossing over might have occurred in her, she was homozygous recessive, so the recombinants would also all be homozygous recessive. That's why we can assume this. Now, if the genes are not linked, we expect the phenotypes to come out in quarters, like we've been saying. If you're sitting there wondering, why does she know that it's quarters? I just did a dihybrid cross between these two genotypes, and then I counted the phenotypes. I'm going to give you the data. You're going to decide linked or not linked.
778 smooth wings, red eyes. 162 smooth wings, yellow eyes. 158 wrinkled wings, red eyes. 785 wrinkled wings, yellow eyes.
Take a minute and decide whether you think they are linked or not linked. You got it right if you said that these genes are linked. They did not distribute the way that we thought they would. What you can expect from me is some practice problems and quizzes sets to come to you. The quizzes check themselves automatically, but the practice problems, after you do them, I'm going to submit a video of me going through the answers because I feel like that's the most fair thing that I can do for you. The final thing I have to say is bad, and it's that if we have school on Monday, we will probably be taking a test on Monday. So that means today we finished learning. Tomorrow we're going to review. Friday we're going to try to take the online progress check. And then Monday we're going to try to test. We'll see what happens. Do your best to ask anything that you want to know the answer to, you can message on Instagram. Please don't comment on the video. I definitely won't notice. Or you can post in the Facebook group, and I'll notice that too. Bye, guys.